Good evening. Welcome to the 2017 Jefferson Pilot Lecture. The Jefferson Pilot Professorship is an endowed award established in 1981 through the generosity of the Jefferson Pilot Life Insurance Company. ACC fans will remember the commercial, Sail with the Pilot. It is the highest and most singular honor North Carolina Wesleyan College bestows upon its faculty. The honoree is nominated by a committee of faculty and appointed by the president. At this time, I'll ask Provost Evan Duff to introduce our 2017 Jefferson Pilot recipient.
Good evening. I told Dr. Louch I was probably going to be more nervous than her. Dr. Heather Louch joined the North Carolina Wesleyan College faculty in 2002 and became an associate professor in 2008. During her time at Wesleyan, Dr. Louch has excelled in her teaching and commitment to students. Evidence of this includes her winning the Exemplary Teacher Award in 2014, as well as creating numerous biology courses, creating the Bachelor of Arts degree in biology, and creating the biomedical major. Dr. Louch also started the annual Student Symposium in 2004, which still continues today. In addition, Dr. Louch has provided a great deal of service to the college. This includes leading workshops for faculty and students, and for serving as the program advisor for the GlaxoSmithKline Women in Science Scholar and Scholarship. I now present Dr. Heather Louch, who will provide a lecture on Western lifestyle, Western diet, a body and microbiome out of sync.
Thank you, everyone, for coming tonight. Before I get started, I just want to say, point out a few people that are in the audience that I'm very appreciative to have here. So first of all, I do have some former students here, and I appreciate their coming tonight. That means a lot to me. And then second of all, of course, my family is here. My mother, Janice Metz, has traveled here from Charleston, South Carolina. And then my mother and father-in-law, Judy and Pete Wozniak, have traveled from Cleveland, Ohio. And then, of course, my children are here, Damien and Dominic, and my husband, John. And with them, I greatly appreciate their patience as I've worked through this process of bringing this to you tonight. There's been a lot of, Mommy, can you play? No, I got to do something right now. So, when's your Jefferson Pilot going to be done? So, not long now. All right, so with that, I will get started. As you can see, the title tonight, Western Lifestyle, Western Diet, a Body and a Microbiome Out of Sync. So before the advent of agricultural and pastoralist societies, ancestral humans' dietary choices were limited to minimally processed wild plant and animal foods. The domestication of plants and animals, followed by their cultivation, occurred approximately 10,000 years ago. This resulted in novel foods that became staples for many human populations. During that transition period, many hunter-gatherer populations continued to exist and subsist on wild-caught foods. It is certainly true that hunter-gatherer societies still exist today and are actively studied by anthropologists. The Tsimane of Bolivia in the upper right-hand corner, the Bajau of Malaysia, a sea-dwelling society, and the Hazda of Tanzania, across the bottom left, are just a few of the hunter-gatherer societies in the world today. Fast forward to humans living in industrialized societies today, and we acknowledge that there have been many additional dietary changes. The human diet now includes foods that are in many cases far removed from the wild state. Readily available, processed, calorie-dense foods are the norm. Refined sugar, for instance, has been processed from plant matter into a form devoid of other nutrients and fiber. It can be used for additions to manufactured or home-prepared foods. Processed fats are also a common food ingredient, taking the form of lard, shortening, or oil. Recent advances have increased the variety of seed oils available for consumption, and we see these across the bottom of the slide. These calorie-dense food ingredients are often added to grain-based ingredients like corn and wheat to form the basis of many of the prepared foods that we find at the grocery store or in restaurants. With foods of convenience so ubiquitous and relatively low in cost to the consumer, it is very easy for individuals in industrialized or westernized societies, such as the United States, to allow these foods to form the basis of our modern diet. This so-called Western diet is thus best characterized as a calorie-dense diet with little variety. It is comprised of a significant proportion of foods well removed from the natural form. In other words, foods that are highly processed and consequently often low in fiber and high in refined sugar and processed fats. Artificial preservatives and colorings are often added. So in addition to the Western diet, there is also a Western lifestyle. Most would agree that few of us engage in the amount of daily exercise that would occur were we living in a hunter-gatherer society. In fact, humans in westernized societies spend a lot of time sitting indoors as we work, pursue entertainment options, or engage in hobbies that are primarily indoor activities. This lifestyle is enabled by affordable artificial lighting, which has allowed us to experience prolonged hours of indoor illumination and just an average of 1 hour outdoors each day. If you've ever stopped to consider the way the light changes throughout the day, you may not be surprised to learn that most interior lighting is out of sync with the natural light stages of the day. Ideal interior lighting would change throughout the day to allow us to remain in sync with our biological rhythms. While any amount of artificial light in the evening can disrupt natural biological processes that promote sleep, blue light in particular suppresses the natural rise of the sleep hormone melatonin. In westernized societies, blue light emitting electronic devices are commonly used into the evening hours. The frequent result is difficulty falling asleep at the desired time. Even without blue light exposure in the evening, the artificial interior lighting allows humans in westernized societies to modify sleeping behaviors by choice due to personal habits or work schedules. Performing night shift work on a permanent or rotating basis is common in industrialized societies. Many of these workers shift back to normal patterns on their days off. The shifting sleep schedule is not limited to shift workers, as many people change their sleeping patterns on the weekends in order to accommodate social outings or just to sleep in for a change. No matter the reason, a shifting sleep schedule results in a jet lag type paradigm, which has negative consequences on human biological rhythms. Here we see one artist's rendition of what it feels like to have jet lag. So westernization has been identified as a leading culprit in the new era of disease that we see today. This new era is marked by chronic, metabolic, and immunological diseases, such as, and this is not everything on there, and not all these are on there, diabetes, obesity, cardiovascular disease, food allergies, depression, cancer, and inflammatory bowel disease. These diseases are just some of the negative health consequences. Others are shown on the slide that became prevalent following industrialization. Certainly, multiple factors are involved in the rise of these Western diseases. However, there is a growing appreciation for the contribution of the microbiome, an individual's native bacterial assortment, to the complexity of these disorders. The bacteria in the human body and the genes they possess, the collective microbiome, are known to be important in educating the intestinal immune system. From the early stages of life, specific bacteria that are maternally and environmentally acquired are required for immune development. It is therefore important to consider whether alterations in our physical environment in recent history have led to a shift in our personal bacterial communities. The relative role of our microbiome in human health and disease and its adaptive evolution to westernization will likely hold important identifiable cues to the understanding of modern disease. As we begin to look at some recent research on health impacts of westernization, there is an important context that I wish for you to keep in mind, as it is the basis for some of the studies that we will consider today. Virtually all animal species possess body fat as an energy storage adaptation, permitting survival when the food supply is intermittent. Unlike modern humans and domesticated animals, animals in nature successfully live on an eating schedule involving intermittent energy intake, meaning that they may go extended periods of time without food. Many modern humans consume calories regularly throughout the day, often beginning within a few hours of waking and ending within a few hours of going to bed at night. Scientists therefore wondered whether the constant fed state of many humans in modern urban societies, in conjunction with the Western diet, may be contributing to the prevalence of metabolic and inflammatory diseases. Scientists have utilized a mouse model to try to tease apart some of the Western elements that are external to any mammal, including diet and light-dark cycles, from the internal elements of the organism, such as obesity, activity levels, physiological elements of metabolism, and the gut microbiome. As you can imagine, this is not a simple feat. For dietary studies, an inbred mouse strain called C57L6J, pictured here, is commonly used. They are considered to be a general multi-purpose model and are useful for exploring diet-induced obesity and type 2 diabetes. These mice are reported to have mildly elevated blood glucose and impaired glucose tolerance, making them ideal for metabolic studies. To assess the impact of a Western diet, mice were divided into two groups and were fed either a normal chow diet, indicated by the letter N, or a high-fat chow diet, indicated by an F, with the high-fat diet representing an element of the Western diet. This 60% fat diet, which is high even by human dietary standards, is the gold standard in high fat for mouse experiments. To assess whether the constant fed state has any impact on Western disease, mice in each dietary group were split into subgroups based on when they were permitted to feed. On the right side of the slide, we see a bar in white and blue that defines the 24-hour day, with 12 hours of light and 12 hours of dark. Next, look at the long yellow bar above that, which spans the 24-hour day. This shows unrestricted or ad libitum access to food. Mice placed in this feeding group could thus eat when they choose at liberty. We see this ad lib feeding arrangement represented by the letter A for some mice in each dietary group, normal or high-fat chow. The remaining mice had their feeding restricted to an 8-hour time frame during their active phase, which for mice is at night. And we see that here as a much smaller yellow bar below the night cycle. Mice on this time-restricted feeding regimen, represented by a T, were from one or the other dietary groups, normal or high-fat chow. Mice were subjected to their respective normal chow or high-fat diet with either ad lib or time-restricted feeding for a period of at least 100 days. Mice in the two dietary regimens had the same caloric intake despite the different nutrition composition of the diet. Let's consider the experimental outcome of these four groups of mice. While the average daily calorie intake for individual mice in all four groups was equivalent throughout the 18 weeks of the study, the mice fed a high-fat diet with a restricted feeding time, the FT group, were protected against the excessive body weight gain experienced by mice on the same high-fat diet feeding ad lib, the FA group. The top line of the graph shows the body composition of the mice in the FA group, while the line under that shows the FT mice on the same high-fat diet with an 8-hour time restriction. Again, keep in mind that calorie intake and diet composition are the same between the mice represented by those two top lines. Note the size difference between the fat-fed mice. These are both fat-fed mice, as well as the associated body compositional differences. Note on the graph to the right that while lean mass is approximately equal, the tan color in the middle of each bar, the fat mass was much greater in the FA mice fed high-fat diet ad lib. 70% more fat deposits were measured in the ad lib mice on the high-fat diet than the time-restricted mice on the same diet with the same caloric intake. An analysis of the livers of the mice showed a fatty liver in the FA high-fat ad lib diet group, whereas all other mice, including the FT time-restricted high-fat fed group, showed a healthy liver. So at this point, the researchers concluded that the time-dictated feeding was able to reprogram some of the molecular mechanisms of energy metabolism along with body mass regulation and body mass distribution. But they were curious about other factors relating to body metabolism. The clearly obese mice were hypothesized to have other hallmarks of obesity that are seen in humans, namely elevated glucose levels and elevated insulin levels. But what about the mice on the same diet with time restriction? Would they be normally sized mice that have metabolic disorders? Or would they compare better to the mice fed on healthier diets? So as a quick overview, in a healthy individual, insulin is a hormone that is produced in response to intake of a meal, starting at the green box. We see that insulin is produced in higher levels when more glucose is present in the bloodstream. When this insulin hormone is produced, it signals to the cells in the body that glucose is available for uptake. When cells respond properly, glucose levels drop, the turquoise box. Homeostasis will be achieved, and blood sugar will return to normal levels without the need for high amounts of insulin. In individuals with systemic inflammation, insulin levels will be elevated and the cells will not respond as readily to the insulin present. In these individuals, it is difficult to normalize glucose levels in the blood. The condition is a precursor to type 2 diabetes. So researchers examined these mice on a high-fat diet for other signs of metabolic dysfunction by looking at their insulin and glucose levels in response to a glucose challenge. To do this, glucose was injected into the abdominal cavity of the mouse, and blood glucose was measured in intervals of 30 minutes for 2 hours. While the FA high-fat ad lib mice showed hallmarks of glucose intolerance due to poor diet, see the top line on the graph to the left, the FT high-fat time-restricted cohort did not. Instead, these FT mice showed both glucose tolerance and insulin levels comparable to the control NA ad lib normal chow-fed mice. From the figure on the right, we see that insulin levels are elevated in the FA ad lib high-fat diet group before glucose is even administered. So look at the minus signs across the bottom. Following a glucose challenge, now looking at the plus signs across the bottom, we see that the levels of insulin are extremely high in the FA ad lib high-fat diet group in comparison to all other groups of mice. Once more, look at the FT mice on the same poor diet, but permitted to eat only in an 8-hour window each day, and note insulin levels similar to mice on the healthy diet. Impressed with these results, in an expansion of the study, researchers wondered if similar effects would be seen if mice on the same high-fat diet were granted a more relaxed feeding window. So rather than 8 hours, what if we say they can eat for 9 hours, or 12 hours, or 15 hours? So we see these along the left, plus the very top, we also see the high-fat diet ad lib group again as well. So those are the first 4 on the left side. In addition, researchers were curious. What would happen if the feeding restrictions were removed on 2 sequential days every week? This setup was designed to mimic a behavior seen in humans, which is often that the diet goes out the window on the weekend. So in this experimental group called 5T2A, mice fed a high-fat diet were kept in a 9-hour time-restricted feed for 5 days of the week, and then permitted to feed ad lib of the same high-fat diet for 2 days of the week. This, in essence, permitted scientists to ask whether time-restricted feeding has a legacy effect that can override occasional interruptions by ad lib feeding. The groups on the right were fed the normal chow, which in this study only included an ad lib group and a 15-hour time-restricted group. So even with the equivalent amounts of food consumed, it was quite apparent that the mice fed a high-fat diet. If you have longer feeding times, you will have a larger increase in weight. In addition, the 5T2A mice, which get the weekend off from their restricted feeding time, had a body weight gain falling in between the 9 and 12-hour time-restricted groups. The mice on a 15-hour time restriction were moderately obese, and as such, tended to show metabolic indicators like blood cholesterol, glucose levels, and insulin levels that were better than the FA high-fat ad-lib feeding group, but not levels considered to be healthy. Because gains in health standing are often reflected in improved endurance and motor coordination, these mice were examined for their endurance on a treadmill. All right, so not surprisingly, the obese FA mice, which fed ad lib on a high-fat chow diet for 12 weeks, did not last as long on the treadmill as the NA group eating normal chow ad lib. What was surprising was that all mice fed on a high-fat diet with time restrictions performed as well or better than the NA normal chow ad lib group. The mice on the high-fat 9-hour time restriction endured the treadmill for longer than any other group at a full hour longer than the NA control group. Likewise, the mice fed the same way as the 9-hour group, but who got to feed ad lib on the weekend, were pretty impressive as well, enduring the treadmill for 45 minutes longer than the NA control group, and nearly as long as the 9-hour group that do not get the weekend off. Mice were also tested for their motor coordination using a device called a Rotarod. You'll notice in a moment that the Rotarod has a sensor pad below the mouse which stops the time when the mouse falls off the rod. Let's watch a short video to see what this protocol entails for the mouse. The results shown here from the Rotarod coordination test indicate better coordination for mice that are maintained on a time-restricted diet. These specific results are for mice maintained on their respective eating regimen for 38 weeks, rather than the typical 12 to 15 week period. The comparison here is between the high-fat diet ad lib group labeled FAA for the extended time on their diet, and the high-fat 9-hour time restricted group labeled FTT for their extended time on their respective diet. So thus far, the researchers have been unable to explain these coordination and endurance differences. They examined forelimb muscle strength, muscle fiber size, and muscle composition between the different mouse groups and found no differences. There was no difference in total mouse activity across the different groups, and therefore there's not seemed to be a training effect. They also investigated whether mice with less endurance were running out of muscle fuel. However, glycogen, the muscle fuel, was higher in ad lib fed mice. As we just saw, ad lib fed mice were the ones that did not perform as well in the coordination or endurance testing. The constant access to a high-fat diet in ad lib feeding mice made them lose their nocturnal feeding rhythms, which is their desire to eat mainly at night. These circadian feeding rhythms were out of sync. This, in combination with the poor diet, predisposed these mice to obesity and metabolic diseases. A time-restricted feeding regimen acted as a reset button, preventing a high-fat diet-induced disruption of metabolic function. It appears that lifestyle modifications involve restricting meals to a specific time frame during the active period of the day can override the detriment of a so-called bad Western diet. The other factor in play here that has not been addressed on this slide or in the research I have shared with you is the microbiome. We know enough about the microbiome to realize that these microbiota are significantly important in carrying out metabolic functions in the body and may play a role in obesity. It was first reported in 2006 that gut microbiomes of obese humans and mice differed from lean controls. Genetic analysis of obese-associated microbes found enrichment in genes associated with lipid and carbohydrate metabolism. However, the relationship between such genes and metabolic activity is not well understood. The gut itself is in a constant flux based on feeding and fasting cycles, and that impacts the quantity of nutrients, the pH, and various metabolites produced. However, it was unknown whether the gut flora would be similarly dynamic. Based on the studies on time-restricted feeding, the next objective was to determine whether the gut microbiome is affected by cyclical fluctuations in feeding. Also, mice eating normal chow restrict most of their feeding to their nighttime active phase. So how would this impact cycling bacteria as well? There are 4 groups of mice that the researchers were particularly interested in due to previous findings indicating differences between lean and obese individuals. I'll show you data for 2 of those groups. But before we look at that data, you need to understand that the data is acquired by analyzing RNA. The data measures reads, which in simple terms tells you how frequently that RNA sequence appears. The RNA sequences analyzed are unique and therefore specific for individual bacterial species. Also, when we look at the data, we will be looking at both cycling of a particular bacterium on the line graph and total amounts of the bacterium on the bar graph. In this study, the ad lib and 8-hour time-restricted feeding groups on high-fat or normal chow diets were again used. So the first group of organisms we will consider are the lactobacilli. Several species have been studied extensively, and lower levels of these organisms appear to be protective against metabolic disorders associated with obesity. Starting with the bar graph on the right, we see that the FT high-fat diet time-restricted mice have a much lower level of Lactobacillus species reads compared to either ad lib cohort. On the graph to the left, while the cycling pattern of bacteria within each different mouse cohort is slightly different from one another, amounts of bacteria do seem to be greater in the light cycles, which are shown across the bottom of the graph as white blocks. In the second group that we will focus on, the Lactococcus, studies have shown a direct correlation between a higher level of this organism and increased body fat percentage in mice. Looking at the bar graph on the right, we see that the prediction of higher amounts of Lactococcus species as body fat increases is supported by the data, with higher levels seen in the obese FA high-fat ad lib feeding mice and lower levels in both the leaner FT and NA mice. Curiously, the bacteria in the FA mice, seen in yellow on the left, appear to be somewhat cyclical. When microbial diversity as a whole was considered for any specific time point during the day, the ad lib feeding mice on a normal chow diet always had more variety of microbes present, as seen by the different colors in the top panel. And that diversity was always higher during nighttime feeding cycles, which is not well represented on this pictorial summary. In contrast, the FA mouse cohort, which lose nocturnal feeding patterns, did not contain as much diversity of microbes. There were very few cycling microbial groups, which are not shown on that image. They also had gut flora predominated by microbes previously found to be associated with obesity in humans and mice. Researchers had hypothesized that the FT group would make the gut microbiome highly dynamic, like the NA control group, but this was not the case. Like the FA mice, the FT mice had low microbial diversity for each time point in the day. They did have more overall diversity in comparison with the FA group, but microbial gut diversity paled in comparison to the NA group. Obesogenic organisms were reduced, and obesity protective organisms were increased. So subtle differences in the gut flora of the two high-fat diet groups must therefore be contributing to their overall physiological differences. The implication is that host diet is a strong dictator of which microflora are found in the gut. The high-fat version of a Western diet may lead to obesity and to a microbiome that differs in relative abundance and in the cyclical oscillations of the gut microflora. Feeding restricted to the active portion of a mammalian day can both correct for obesity and bring microbial cycles and key organisms more in line with those seen in a normal diet, thus preventing the detriment otherwise seen in animals consuming a high-fat diet. While the oscillations seem to be diet and food timing related, whenever biologists start to speak of cyclical oscillations, the term circadian rhythm inevitably comes into the conversation. A circadian rhythm refers to the approximately 24-hour cycling internal clock that occurs in living beings. The clock can be influenced by external time givers called Zeitgebers, such as meal timing and physical activity. However, the strongest regulator of the circadian clock is from light cues received through the retina of the eye. Light-sensitive receptors direct the light to a region of the brain where the central or master biological clock is located. Light cues are used regularly as a reset mechanism, allowing adaptation to seasonal changes in dark-light cycling. When light is received into this region of the brain, signals are sent through hormones and the nervous system to other organs and cells in the body. These signals dictate rhythmic expression of some genes in the body and help to keep the other cells in the body aligned with the master clock in the brain. There's also evidence that organs and cells can be out of alignment with the master clock in the brain, cycling differently. For instance, if the human diet disrupts a gut circadian rhythm that is now out of alignment with the master clock, this could promote the metabolic health problems seen. These, in turn, would be exacerbated if the gut microflora attempt to follow two different clocks, the master clock through gene signals and the food clock through dietary intake and timing. Going back to the light element of the circadian rhythms, researchers were curious as to how the Western lifestyle, namely extended light exposure, may be having detrimental impacts on health. While artificial lighting allows people to extend daytime activities into the night, human sleeping quarters are often bathed in the glow of various electronic light-emitting devices, from alarm clocks to power strips to even television sets. Sometimes a street light may shine into our bedroom window. This dim light glow at night is most commonly referred to as light pollution. So scientists wanted to see whether there is a link between altered light cycles and metabolic disorder. This time an albino mouse strain called Swiss Webster was used. Other than being albino, it is a standard mouse strain with no predisposition to obesity or metabolic disorders. Mice were housed in one of three lighting conditions, which are represented by the clock faces. The first group, called LD, was given a standard light-dark cycle with 16 hours of light and 8 hours of darkness. The dark night period is shown as the dark third of the clock face. Another group, called LL, seen on the far right, was given 24 hours of continuous lighting. The last group, called DM, was given an in-between state where there were 16 hours of light and an 8 hours of dim light, with the dim light nighttime shown as the gray third of the clock face. Inclusion of the all-light LL group allowed researchers to study the metabolic impact of a desynchronized circadian clock where the biological clock inevitably runs free. The dim light at night, DM group, models the impacts of nighttime environmental light pollution. Mice in DM and LL conditions were hypothesized to have altered metabolic features in comparison to the control LD mice. Specifically, mice in these groups lacking complete darkness were expected to have reduced glucose tolerance and increased body mass. Additionally, the LL mice receiving no type of darkness were predicted to have motor arrhythmicity due to circadian disruption. As we can see from the top two lines on the top graph on body mass, the LL and DM mice lacking complete darkness gain more weight over the study period. Because a well-synced body should restore glucose levels to homeostatic levels more quickly, it is logical that the LD group receiving complete night darkness recovered more quickly from a glucose challenge, as seen in the bottom line on the bottom graph. In contrast, the top two lines of the same graph are for the DM and LL mice, which were both not well synced in their circadian rhythms due to their respective nighttime light disruptions. It is therefore not surprising that these mice did not respond as effectively to a glucose challenge. This graph looks at mouse activity, and the higher any individual bar, the more activity at that time point. Note the black or gray bar across the top of panel A and B, which indicates the length and type of nighttime darkness. Panel C is, of course, lacking any night darkness. Activity monitoring shows that LD mice receiving a daily standard dark night period limit majority of activity to their active phase in the dark. In contrast, mice that never receive any darkness, seen in panel C, do not sync their activity with any sort of rhythm, while mice that are receiving a dim darkness, seen in panel B, do maintain rhythm to their activity. These observations are supportive of the hypotheses that were put forward. Returning to the metabolic harm observed with inadequate darkness, the next question was whether it would be possible to use a set feeding schedule to entrain the circadian system and eliminate some of that harm. Mice were all fed the same normal chow diet and assigned to one of three food access groups: ad lib feeding, abbreviated as A, dark restricted feeding, abbreviated as D, and light restricted feeding, abbreviated as L. These mice were assigned to either the LD or DM groups previously described. On the graph, we see the three feeding groups across the bottom, where F simply stands for food in this case. On the right side, we have the dim light at night, and on the left side, a completely dark night. The results tell us a few things. First, even with a healthy diet, feeding when you should be sleeping, both FL groups, favors an increased body mass. Second, mice who eat ad lib but receive a complete dark night, FA, seen on the left, are protected against the weight gain experienced by mice in a night with light pollution, FA, seen on the right. So restriction of food to the mouse active period during the dark cycle, even when darkness wasn't complete, bestowed a protective effect on mouse weight gain. The finding that meal timing has major effects on metabolic and physiological outcomes both in a light-disrupted and a diet-disrupted mammal has led to the conviction that in choosing food, it is not only important to consider nutritional value, but also its timing. Feeding and fasting cycles, in addition, of course, to light, do appear to be potent timing cues that align circadian rhythms that may have become unsynchronized. And when food intake is unaligned with the circadian clock, the clock may be put in disarray. When clocks become disrupted or unsynced, as is the case in human night owls, these individuals are at higher risk for developing poor health outcomes than individuals who are early risers. One explanation is that circadian rhythms may have been uncoupled from the central clock in the brain due to late night light exposure and eating habits. A number of studies have demonstrated that shift workers have higher rates of cancers, cardiovascular disease, obesity, psychiatric disorders, and neurodegenerative diseases. So we've seen some studies that suggest that the gut microbiota are following their host circadian clock. The mammalian microbiota are pivotal to a variety of host functions, some listed here, all of which show some element of circadian control. So how does this gut microbial ecosystem follow the circadian oscillations in the host? Is a properly set host clock required for the microbiome to undergo any rhythm? And what is the consequence if the host and gut microbial communities are misaligned? In a final set of experiments with Swiss Webster mice, the same albino mice used in the light-dark study, researchers wanted to explore just how connected the gut microbiota are to the circadian rhythm in the host. To do this, jet lag was induced in the mice by advancing the light cycle 8 hours, holding the mice there for 3 days, and then returning them to the original light-dark cycle. For example, mice currently experiencing nighttime from 10 a.m. to 10 p.m., because this is a lab after all, would next experience nighttime from 6 p.m. to 6 a.m., and then would be shifted back to the 10 a.m. to 10 p.m. nighttime. This back and forth was repeated every 3 days for 4 weeks. Despite the term jet lag, this more closely mimics a scenario of regular shift work, where one must switch regularly between day and night work. Here is what I imagine that these time-shifted mice may have felt like. So after 4 weeks of this constant back and forth shifting, mice were returned to the starting light conditions and analyzed the next day. Not surprisingly, the jet-lagged mouse cohort was a bit mixed up when compared to non-jet-lagged mouse controls. Beginning at the top left, we see that while quantity of food intake, shown in black, was similar between the two groups of mice, the jet-lagged mice show even amounts of food being eaten in the dark and light phase, which is inconsistent with a well-synced nocturnal animal. Additionally, seen at the bottom right, the rhythmicity of Ruminococcaceae in the jet-lagged mice, shown in red, was basically absent, and the levels of these species were quite low in comparison to the non-jet-lag control. Lower levels of this family of bacteria are associated with obesity. This is just one representative of the impact on cycling bacterial groups. Results like this were the norm for the jet-lagged mice. Because of the microbial imbalances and loss of proper microbial oscillation in jet-lagged mice, scientists asked whether the metabolic imbalances that are seen with altered circadian rhythms in the host could be attributed to the microbiota. Using the 60% high-fat Western diet to mimic the dietary habits that predispose humans to metabolic syndrome, the mice were split into two groups, with some receiving the jet lag induction and others not. Finally, because they wished to determine if the microbiota were contributing to the metabolic phenotype, an antibiotic cocktail was administered to a subset of both the control and the jet-lagged mice. Antibiotics either kill or prevent growth of bacteria, such as the gut microbiome. Remember, all these mice have been fed the high-fat diet. Comparing the top red line in both graphs with the solid black line on both graphs, we see that mice experiencing jet lag while feeding ad lib exhibit glucose intolerance and enhanced weight gain compared to non-jet-lagged mice. The red line shows the jet-lagged mice. The graph to the left is looking at weight gain, while the graph on the right is looking at glucose challenge. Now focusing on the dotted lines on both graphs, where antibiotics were administered to the jet-lagged mice in blue and the control mice in black, we see that the mice undergoing 10 weeks of jet lag while eating a terrible diet ad lib can overcome the metabolic outcome of obesity and glucose intolerance when administered antibiotics. Again, focus on the blue dotted line on both graphs. These results suggest that the microbiota were actually contributing to the metabolic disarray. When antibiotics removed or reduced the microbes, the impact of jet lag was similar to that seen in the control group without jet lag. Scientists wanted to know if they could recreate the metabolic imbalances from the jet-lagged mice through the microbiome alone, and they were successful in this endeavor. The experiment involved performing a fecal transfer of either control or jet-lagged gut microbiota from the control or jet-lagged mice into germ-free Swiss Webster mice. So germ-free mice do not have their own gut flora. The results at the 30-day time point show that the recipients of the jet-lagged microbiota displayed enhanced weight gain and body fat in comparison to the control microbiota recipients. They also showed glucose intolerance. So how do these mice jet lag experiments translate into humans? A very small-scale human experiment was performed with 2 human volunteers who agreed to cross multiple time zones through an 8 to 10-hour flight-induced time shift. In the days prior to departure, multiple human fecal samples were obtained for a baseline. The key finding from these samples was that the cyclical configurations of the microbial community, seen as the little cycling waves there, were repetitive and occurred at the exact opposite times of the day compared to the mouse microbiota, which is consistent with the diurnal versus nocturnal behavior of the different mammalian hosts. Human subject fecal samples were also obtained 24 hours prior to jet lag, 24 hours into jet lag, and following recovery from jet lag, which was 2 weeks later for this extended trip. These microbiota were introduced into germ-free mice. So there are 3 different time point transfers of gut microbiota that occurred into mice. Human gut flora from before the trip, human gut flora from during jet lag, and human gut flora after jet lag has been recovered from. So mice receiving the microbiota from a jet-lagged human, which is seen as a red line on each of these graphs, displayed enhanced weight gain, shown on the graph to the left, and higher blood glucose levels after challenge, which is shown on the graph to the right. These previously germ-free mice recipients also accumulated more body fat when receiving the jet-lagged human microbiota. So what are the implications? This suggests that human microbiota undergo circadian oscillations, and that circadian misalignment in humans is associated with microbial imbalances that may contribute to metabolic imbalance. It suggests some level of crosstalk between the host and the microbiome in that the metabolic imbalances from the jet lag and poor diet were able to be recreated in the germ-free host that simply received the microbiome of the other mouse. So the point today is not to vilify fat. High fat just happens to be part of the Western diet that this recent research has focused on. In actuality, the aspect of the Western diet that is considered to be among the major triggering factors of metabolic impairments is the increased fat-to-fiber ratio. Thus, it is not necessarily believed that the percent intake of fat in our diets have changed as much as it is that we are not eating adequate fiber in our Western diets. An examination of current hunter-gatherer populations does demonstrate that we in the Western world eat significantly less fiber. Recent studies also support findings that there has been a loss of microbial diversity in industrialized human populations compared to populations using more traditional subsistence patterns. Microbial ecologists have even expressed concern about extinction of unique gut microbiota from humans in industrialized societies and have questioned how many of the metabolic, inflammatory, and immune diseases today could be attributed to loss of gut flora diversity. The technological developments of modern society have allowed for advances that have changed our diet, environment, and lifestyle in profound ways. Few would argue that these changes were not beneficial. However, in considering human health, it is clear that there is a downside to westernization. The chronic, unhealthy states seen in westernized societies are rarely present in native populations. Persistent, subacute, chronic, low-grade inflammation pervading everyday life is now a commonplace Western notion. The intestinal microbiota and its byproducts may not be the cause and the cure to every modern-day disease, but these bacteria are not passive bystanders either. Evidence suggests that there is a role for the mammalian gut microbiome in modern-day disease, and that humans have the opportunity to help direct this microbiome through our dietary and lifestyle choices. Thank you. I'll be happy to take any questions you may have. Yes, Mary.
Were there any studies done that used the time-restricted mice, but then also had intervals that they could be fed in that time-restricted period? Or were they all fed ad lib within their?
They were ad lib with, that's a good question, they were ad lib within their time period. So food was put out for 9 hours and then removed. Yeah, but there was no like punctuated within that 9-hour period, for instance. So, yeah, good question. Others? Yes, Danny.
Basically, when you talk about high fat,
Yeah.
Apparently they had to have some other nutrients. What percent of their diet was fat when you say high fat?
It was 60%.
60%.
Yeah, yes, yes. Other questions? Yeah, Julian.
Historically, research has been largely performed on males, a variety of species.
Mm-hmm.
However, a lot of agencies and the federal government have been promoting research on women as well.
Mm-hmm.
Do you know if the studies that you looked at had any, or looked at any association between gender and obesity or glucose intolerance?
These studies did not, and there was a recent paper that I did not incorporate. I didn't get a chance to read it thoroughly, but there is a recent study that does look at women versus men and looking at not the time restriction, but looking at the microbiota in particular to see if there's following that same trend. But yeah, so that we are heading. It's a very this field is sort of very much in its infancy. The microbiome, all these research is not very far. I mean, there's so much more to do, and it's generating tons of data that they have to pour through as well. So, yeah. Other questions? Yes, Danny.
Related to that, did they say anything about the gender split of the mice? Did they say X number of males, females, anything like that?
Some of the studies, the light studies, were all males, and the other studies, I don't recall them mentioning gender split, so I guess I would deduce they're probably males as well since they didn't specify. It seems like they tend to use the male mice for these experiments when they have a choice. So, good question though. Yeah. Others? Yes. They were, it was like a milk fat. It was a fat from milk, yeah.
Highly saturated?
Saturated fat from milk, yes. Saturated milk fat. Any other questions?
So am I gathering that you might say it's not just the intake and the quantity of fat that you're having in your diet, but when and how much dark sleep you get?
Yes, yeah.
Change those patterns, maybe we would all be nice.
It seems that way. I mean, that's where the research is headed. It's really looking at that circadian clock and saying how we have managed to mess up our circadian clock with our lighting and our behaviors because of the lighting. And some of that, for some people, you stay up late, it means eating as well. So you eat closer to when you finally do go to bed. And there is some information on a lot of these papers were out of one lab that's at the Salk Institute in California. And they actually have a phone app that they did start tracking humans by this phone app. They have a phone app that allows people to take some baseline data where they're recording what they're eating and when they're eating, and then they encourage them to restrict their intake to sort of set themselves like a 10-hour time frame, for instance, 10 or 12 hours, but they try to push them for a 10-hour time frame that they're going to consume all of their calories or caffeine or anything like that within this 10-hour window, only water outside of that, basically. And everyone in the study they found was different with mice, which was that they would eat, but they would end up not eating the same amount of calories that they normally did when they ate longer during the day. So they would be satisfied on fewer calories. They would just self-restrict, and they actually lost weight as well. And their metabolic markers were improving as well. So, yeah, but that's just kind of a preliminary. It's a phone app that allows them to track data that way. Dan?
Yeah, so popular wisdom tells us that people who work shifts, night shifts for years and years and years, eventually adjust to that, and their circadian rhythm sort of reset. Is that true? And if so, do the gut microbiota in those people also reset?
So the things that I found, and I was having a conversation with someone recently about this as well, but what I found for most people that do shift work is that they don't keep their shift work's circadian rhythm going when they're not at work. They try to readjust to regular life. So around them, people around them are, keeping this normal. So they try to shift back. So I think that's more of the problem. I don't know if they were to keep on that schedule, what they would find. And there's some very interesting possibilities for some poor mice in the future. Good question. Others? Oh, yeah, Mayor.
So I'm a bit concerned now because I saw, as far as I could see from the map, they probably traveled to Israel.
Yeah, that was an Israeli group, yes.
So I'm a bit concerned because I'm planning to go home over the summer. So if I want to minimize my weight gain when I'm flying over there with a jet lag, what do you suggest?
I don't want to make specific suggestions for you because I'm not that kind of a doctor, but some of the research now is looking at probiotics and trying to sort of help the gut flora by pumping in some of those beneficial probiotics. Again, it's early in the research, and to my knowledge, they have not looked at jet lag with that, but they are starting to look at probiotic use and how that affects metabolic outcome as well. So can we replace some of these, with some of these, beneficial organisms? What if we start pumping people with those? What's the outcome? So there's a whole area of research that is looking at, trying to get people's health better by encouraging time restriction as well as going in the direction of probiotics as well. So the other thing is that they say you can do if you're ready to travel is to start to shift yourself a little bit in advance of going, especially when you're traveling that far, because that's quite a shift. They said it takes 2 weeks to adjust to that kind of a shift for jet lag. So, yeah, good question. Others?
I'm just curious,
Yeah.
how did you get interested in this? What pointed you in this field?
So microbiology is the area that I'm genetics, but I'm always interested in microbiology. That's kind of my side passion, and all the labs I've worked in have done microbiology. So I wanted to do something that was a topic that I didn't think many people knew about, but it's out there a lot, that microbiome. So I was curious about, wanting to do that. But the other part of it is that for several years I have used some intermittent feeding strategies myself, so intermittent fasting, and then about a year ago I started to do some of this on my own, this time restrict, where I just restrict myself to some time during the day and I don't have calories. So I was curious about the actual scientific literature and what that would show as well, because I do find more energy and I do find a lot of those things personally, but I don't have the science to back it. It's all personal experience. So I was curious about that. So I was actually very excited when I finally figured out that the term for this was time restricted, because then I started hitting on all of these current research articles. And I was really excited to see that it linked to the microbiota. So good question. Thank you. Any others? Well, thank you, everybody.
Great job, Dr. Louch. Molly has suggested a pop test, but I said, let's pass on that. So those eligible to receive this Jefferson Pilot Award are tenured members of the faculty who have clearly distinguished themselves in ways that set high standards of excellence to which the whole community can aspire. They are held in high regard as teachers, scholars, and members of the Wesleyan community. Portraits of the Jefferson Pilot professors are displayed in the lobby of the Brazel Administration Building. As I read the names of past recipients, please stand and remain standing until I've completed this list. Actually, 7 of these were professors when I was a student here. Dr. Leverett Smith, Jr., Dr. Douglas Goodwin, Dr. Richard Watson III, Dr. Kenneth Finney, Dr. David Jones, Dr. Linda Flowers, Dr. Paul Degategno, Dr. Alan Johnson, Dr. Dale Theron, Dr. Mary Lou Steed, Dr. Barbara Perry Sheldon, Dr. Marshall Brooks, Mr. Everett Adelman, Dr. Jay Quinnen, I think Jay's here, Dr. Steve Farabee, Dr. Vivian Anderson, Dr. Fred Grissom, Dr. Erica Kozel in her bad knee, Dr. Ron Smith, Dr. Jay Stubbyfield, Dr. Nancy Floyd, Dr. Bill Yankowski, Dr. Jonathan Saris, Dr. Carol Lawrence, Mr. Roger Drake, Dr. Kimberly Martin, Dr. Amir McGall, Dr. Fred Sanborn, Miss Kathy Winslow, and of course Dr. Heather Louch. Please give them an applause. You may be seated. It is now my honor to announce the 2017-2018 Jefferson Pilot Professor. This faculty member joined Wesleyan in 2005 as Associate Professor. During his time at Wesleyan, he has excelled in his teaching and commitment to students. Evidence of this includes winning the Outstanding Teacher Award in 2015, winning the QEP Award for a writing exercise in Biology 412, serving as an advisor to the Science Club, and working with colleagues to develop and maintain the Student Symposium. He has demonstrated scholarship with publication of several peer-reviewed journal articles and many conference and professional presentations. This faculty member is respected by his colleagues and just served as faculty of the chair. Most importantly, he knows how to track turtles. So, no drum rolls needed after that. It is my pleasure to announce that Dr. John Temple is the 2017-2018 Jefferson Pilot Professor. If you'd please come forward. Come forward, Dr. Temple, please.
We actually just want to shake your hand.
I'm not ready to follow that right now.
Thank you for being here. We'd like to invite you to a reception. And again, congratulations to Dr. Louch and Dr. Temple. So, thank you.